limitations like donor site morbidity, neuromalformation, fascicle mismatch, scarring, etc. So, currently various types of stem cell alternate sources are being investigated for their application for peripheral nerve regeneration. And reportedly, Schwann cells along with MSCs are most commonly used as the cellular agent for new disorders. But Schwann cells are they have many limitations, like they are very hard to isolate and culture and propagate. Also, their characteristics over time culture. So MSCs the best, and MSC about the beneficial effort not only by differentiating into the Schwann cells, but they also stimulate the native Schwann cell activity, secreting neurotropic factors, and also by increasing the myelination. So the objective of our study was to study the neuroregenerative ability of Patanjali derived MSCs, which will be a promising cell source for transplantation therapy. So the materials and methods of isolation and expansion and characterization of ovine Patanjali derived mesenchymal stem cells and creation of peripheral nerve injury rat model. Our experimental design consisted of 2 groups with 8 animals each, one for control and one for the treatment group. And all experiments were conducted as with the approval from our animal institutional ethical committee. And we had 2 groups. In one group, buffered saline was given as perineural injection after creation of peripheral nerve injury. And in the second group, instead of PBS, we gave 1 million ovine Patanjali MSCs as a perineural application. And analysis of neural regeneration was done based on functional analysis of neuronal regeneration, that is through sciatic functional index, and histological study of the sciatic nerve sections, and study of the muscle nerve atrophy, that is gastrocnemius muscle. The results, we successfully isolated, cultured, and expanded disease. They were characterized based on the presence of surface cell markers like CD 73, 44, and 90, and also the absence of expression of CD 34, and also their ability to differentiate into osteogenic, adipogenic, and chondrogenic lineages. And peripheral nerve injury rat model was created by crush injury on the sciatic nerve, and it led to axonotmesis injury on the left hind limb, and muscle was approximated and skin closed after the surgery. So this sciatic functional index. So it was calculated from the footprint measurement over a period of 6 weeks after the injury. This is the formula for calculation of sciatic functional index. An XFL score of 0 indicates normal, and a score of -100 indicates total impairment. So this is the scores of sciatic function index that we obtained. So we can see that from the 3rd week onwards, there was a significant difference in the treatment group, that is treated with Patanjali MSC group. And atrophy of gastrocnemius muscle was also studied. And Patanjali MSCs had higher relative muscle mass compared to the control PBS group, and we had a highly significant difference in that. Our histological studies revealed in H&E staining, lesser vacuolation and increased Schwann cell proliferation was observed in the Patanjali provided group compared to the control group. In case of mass and trichome staining, although presence of fibrous tissue accumulation was there in both groups, it was comparatively lesser in the Patanjali treated group, and there was increased collagen deposition in the Patanjali MSC group compared to the control PBS, which gives a better functional outcome. So the conclusion is that the comparison of SFI scores revealed significant difference between MSC treated group and the control group from the 3rd week onwards, indicating a better functional outcome. And there was highly significant difference in the average muscle weight between treatment and control group. Lesser vacuolation and increased Schwann cell proliferation was observed in hematoxin and eosin stained sciatic nerve histological studies in the treatment group compared to the control group. And mass and trichome staining also revealed lesser fibrous tissue invasion at the crush site of treatment group compared to the control group, which indicated better tissue healing. So it can be concluded that Avine Patanjali MSCs has neuroprotective effects and effectively promoted the regeneration of peripheral nerves. These are my references. Thank you.
Thank you very much for giving the time and interesting work. Peripheral nerve regeneration is an important subject. So I will take one or two questions and then I will ask the audience. The physiological parameters actually you did not study.
No, sir.
So that was...
We were more concentrated towards a functional outcome after the injury.
Yeah, so, but normally actually, unless and until actually you have physiological parameters coming back to the normal situation, actually, it's difficult to, completely say actually that the regeneration has taken place actually in that sense actually. But yes, I could see that other parameters, mostly parameters and particularly the muscle mass has come back. So how was the study?
It was for 6 weeks.
Okay, yeah.
Thereafter, we started the animals and isolated muscles, histological sections and all.
So any other behavioral study?
No, sir, we only conducted this sciatic function tests.
Okay, any other question, anybody?
Well, dear, nice presentation. So, is it MVC thesis?
It was part of my PhD work, sir. Yeah, it was conducted in Tanvas.
It's only this much is PhD work?
No, sir, it involves much. It is part of that.
Part of that. What was the further?
We actually differentiated these cells into neuronal lineage, and we also provided them also. That also as another.
Why you selected only this Patanjali? Why not other sources of this?
No, Patanjali was easier to access, sir, because in the slaughterhouse also is less invasive.
What
Our lab have already standardized procedure for cultures.
what passage you use it?
We use the third passage.
Third passage. Yeah. So, do you have any study that these were really present there at this site?
Yes, sir. In all the studies, they even tag these cells with the.
That's what I'm really. Is it that you have tagged those cells and you have mapped those or you have imagined them? Or any imaging or something like that?
No, we have not done up to that extent, sir, but of course there are studies, sir.
That's okay, but probably it should have been, very much supported with that really the cells are there, your MSCs are there and they stick there and they do what they want to do there, okay?
Yes.
Otherwise, for sake of results,
Yes, sir. I know it's completed, so further studies are needed, sir. And besides that, infrastructure also matters, sir.
Yeah,
Thanks.
but if you want to take extension of this work further, actually, I think the most important part in this area actually is to also actually use some support. Just only actually giving actually the cells is not going to be something actually which is a clinical translation. So if you look at it from a clinical translation point of view, actually you have to provide some typically if you have a conduit type of thing. In that case, whether it's a crash injury, actually, whether it's a, where you have actually the cut also, actually, sometimes you lose, complete loss, there is no connectivity, actually. Then in that case, actually, you have to provide actually some support where you will also incorporate such type of bioactive. Typically you use NGF, actually, you use many other type of vectors. Yes. So that's just a suggestion, actually. I understand.
Thank you.
It's a small part of work. Yeah. Okay, thank you.
Thank you.
So the next I would just invite now Jayashree Goge.
A very warm good evening to the respected dignitaries and to one and all present over here. I am Dr. Jayashree Gogoi. I am doing my PhD in Animal Physiology, National Dairy Research Institute, Kalyani. So today I am... which one? This is that.
वहीं से मॉनिटर करो ना, वहीं से करो।
फिर चेंज
My title of the topic is Deciphering the Physiochemical and Molecular Properties of the Epinergic System and its 2D and 3D structure in bovine and caprine species. Next. So what is actually epinergic system? Epinergic system consists of first the APJ receptor. The APJ receptor is first and next the APJ receptor is a classic G protein coupled receptor which consists of 380 residues and it is a classic G protein coupled receptor with 7 helical rings. The next two ligands is apelin ligand, which is also an adipoide. It is expressed and secreted and its production is increased during adipocyte differentiation. Next is a newly discovered ligand, which is the apella. It is mostly found during the onset of zygotic transcription in zebrafish. This epinergic system is distributed in various parts of the body and is it plays an important role in various physiological functions. Let's see what are the physiological functions it plays. First, it plays an important role in energy metabolism, that is, it plays an important role in glucose and insulin metabolism. Next, it also plays an important role in food intake. The third, it plays an important role in endothelial cell differentiation, that is angiogenesis. Next, it also plays a major role in cardiovascular system. Also plays an important role in neuroendocrine function and plays an important role in reproduction. It is found in embryo transplantations and also in ovary. So these three ligands have been well characterized in terms of experimental in wet lab techniques in human, but in bovine and caprine species it is very less. So this study has been done with the help of in silico studies. So what is in silico studies? In silico studies of bioinformatics can be defined as a fusion of biology and informatics, which includes applied mathematics, computer science, information technology and statistics. Various research around the world have used mathematical analysis, computational modeling to solve various important biological problems such as protein-protein interaction networks. It also helps to understand the protein structural prediction and modeling of 3D structures of proteins helps in understanding various physiological process, as proteins are important macromolecules which helps in understanding various functional units of life. So in terms of bioinformatics, databases such as NCBI, EMBL, SWISS play an important role. In these databases, there are many functions such as BLAST, which is a local alignment tool which helps in multiple sequential alignment and helps understanding various sequence similarity between the structures. The other bioinformatics tools which have been used, such as ProtPyramid, helps in computing various physiochemical properties with the help of dry lab techniques. Next, the transmembrane domains in the receptors helps in understanding on various protein sequences. Here is one tool called the transmembrane hidden marker burden tool, which helps in calculating the probability of a given stretch of amino acids embedded in a membrane. So what is homology modeling? The modeling is also known as modeling of the 3D structure of a protein from a structure of another homologous protein whose structure has already been previously established. So for this homology modeling, Swiss model is one of the oldest methods used, one of the oldest softwares for 3D modeling techniques, and which uses a rigid body assembly. This rigid body assembly is nothing but it helps in modeling of a known structure with the help of a template. With the help of a template which is derived from the Swiss model repository and helps in modeling of a 3D structure. The phylogenetic analysis helps in understanding the evolutionary relationship among different groups of species and their hypothetical common ancestors. And for this, a software called Megaten software, which uses large interpreter programs for assembling sequence alignments, inferring evolutionary trees, estimating genetic distances and diversities, inferring ancestral sequences. So my objective of this study was to analyze the physiochemical and molecular properties of the apelin APG system in silico and its 2D structural properties. So for the various bioinformatics tools were used to study this ligand, such as protein database was used to acquire the sequences and the sequences were stored in FASTA format. The physiological properties were analyzed using the expressive software. The transmembrane region protein was analyzed using the transmembrane hidden Markov model server. To analyze the protein-protein interaction, the string tool was used. Next, to understand the 2D structure, the SKUAST software is used, and to model the 3D structure, Swiss model software is used. The models which were derived were analyzed using the PyMol software. And the signal server was used to predict the signal peptides in the system. First, the sequences were derived in the FASTA format from all the bovine and caprine species. These are the sequences which was derived from databases such as NCBI and the Swiss Prot. Coming to the results, it was seen that first the isolated pH of the system ranged between 7 to 12, which indicates the basic nature of the peptides, and the ligands were hydrophilic in nature and its receptor was hydrophobic. Coming to the results for the gravy, there were a number of positively charged ratios to range from 9 to 34, and negatively charged ratio ranged from 0 to 34. The instability index ranged from 40 to 85 and was found to be unstable in the test tube. The results were in accordance with the Chang and Paul, who reported physiochemical properties of the apelin system in human, but no reports have been found in bovine and caprine species till date. These are the data which has been found, and the cleavage site for all of those ligands, apelin and apella, was 22 to 23, and there was no cleavage site in the receptor. Here you can see there is a signal peptide in apelin, which is the ligand. There is a signal peptide which is in 23 residues, whereas there was no signal peptides were found in apelin receptor in bovine, and here in caprine similar results were found. The transmembrane region analysis showed that the apelin receptor was seven transmembrane domains were found, whereas there was no transmembrane region in the ligands in both bovine and caprine species. And the apelin receptor was hydrophobic in nature, and the results were in accordance with the Dowd et al., who reported the apelin receptor for bovine, human, rat, mice, and they were hydrophobic in nature. Coming to 12 proteins who interacted with apelin in bovine and caprine. These are the proteins which has been listed. Coming to 2D structural properties, in both bovine and caprine, alpha helix predominated over the beta turn, and then the helical structures. Here first it is shown that the helix was predominated, then comes the sheet turn, then comes the turn and the coil. Coming to the evolutionary structure, it was found that there were 16 disordered regions in the ligands, and 3 best templates were used for 3D modeling as shown by the Ramachandran plot. These are the models which were generated, the 3D structures were generated. And coming to the epigenetic system in the phylogenetic analysis, it was shown that the Capra, Hircus, Saxcrofa, Botsaurus, they belong to the same group. And coming to the conclusions, both bovine apelin in bovine caprine revealed no transmembrane helix, whereas the apelin receptor bovine caprine was composed of 7 transmembrane helices. Secondary structure of apelin receptors revealed alpha helix structure predominated over beta turns and extended turns. 3 best templates for prediction of 3D structure was revealed, and the prediction of the 3D model of the protein in silico analysis is a highly challenging aspect to confirm the spectroscopic data. This report can contribute to understanding of the structure, physiochemical properties, structural motifs, and protein-protein interaction of the apelin system, bovine caprine, for the first time. Thank you.
Thank you. very elaborate work, actually, and is this work published?
No, sir. So now it's been submitted, but it's not published right now.
Okay, it is being submitted. Which journal?
So Journal of Genetic Engineering and Biotechnology.
Okay, so what further actually you are on these, information actually which you have
So
got?
it is just a guiding point for other experimental validations in bovine and caprine species. This whole research will act as a further guiding point for further drug designing or any other experimental validation in bovine and caprine species.
That's the
Yes, sir.
Any other questions? Audience? Audience?
In terms of protein interaction, you said it has receptor interaction, and you did, which docking system did you use?
I use the ZDoc software in terms of protein-protein interaction. Only one ligand I have what I've used, that is I have designed and then I have docked in the apelin receptor. So 1 day or 2 days, that's it.
You mean it doesn't have
Yes, sir, so I could not hear you.
that we brief the first studying the protein and that might
Sorry, I still can't get you. So,
okay, I think we are short of time.
okay, sir.
We can talk.
Okay, sir. Thank you, sir.
Yeah, thank you, Jayesh. Thank you. So next speaker is Preeti Lakhani.
अब समझ गए वो बोलता क्या है इसको क्या बोलता है वी सिंह क्या बोलता है वो समझ गए आप ये तो मैंने नहीं समझा
Good evening, everyone. Myself, Dr. Preeti Lakhani, Assistant Professor Luwas. I am here to present my work. The title is Switching System, which operates automatically based real-time intensity of animal, birds, farm animals in all weather conditions. So, light, it is very essential component in case of domestic animals. There are basically two sources of light, natural, which is available to us, and it is basically cheap. The artificial, man-made, which we use. Now this light act as an exogenous factor which regulates various physiological functions that exist in case of domestic animals. In terms of, it regulates the rhythmicity of various hormones, which regulates production, reproduction, and out of that, behavioral, physiological, and reproductive responses are all regulated with the help of light. So, what's new in my research? Basically, our team has developed a switching system. Now, how this switching system is new from the existing one? These are the switching systems which are actually available in the market. Manual operating, it requires human intervention, on and off button. Timer based, we set the timer and the switching system starts. Motion based, some motion is required, so then the system starts. Command based, your example is Alexa. So our system, how it is different from that system, so we have kept the objective. The objective of our invention is to develop a switching system that operates automatically based on the real-time intensity of animals in all the weather conditions. So two important words are here, that operates automatically and on real-time basis. Automatic means it does not require any kind of intervention, and also real-time intensity, which means it gives the what is the particular temperature during that time. So keep this in mind, the switching system that we develop. So basically it comprises of two sensors and there is a movable flap on it. So two sensors, sensor 1, it has a higher cutoff, and sensor 2, it has a lower cutoff, and movable flap is over the sensor 2. So this is a diagram showing the sensor having a flap, and it also have a LDR system. What is LDR? It's a light detecting resistor. So two types of LDR we are using. One motor, that flap that I'm talking about, it requires a motor. Based on this motor, the flap covers or decovers the sensor 2. So this is the flap which was covering the sensor 2, it moves back with the help of the motor system, and again the bulb gets switched off. So in initial stage there is closing, and in final stage there is opening of the circuit. So relay system 1, closed state, and relay system 2 is the open state based on the intensity of the light. So in conclusion, I will say basically it's arrangement of sensors and flap where no human intervention is required, and it operates automatically depending on what is the condition. And one more thing I need to be mentioned, that the sensor which I discussed before, they all had the light sources above the sensor. Our switching system is having the sensor above the light source. So any kind of disturbance because of cloudiness, darkness, that has also been avoided while keeping in mind of the development of the sensor. So we have gone for the patency of this technology under the project LLM that has been funded to our department. Thank you.
Thank you. Thank you, very interesting work. Any question?
So how much lux you have taken here?
So, 25 and 5 lux. We are actually working on poultry. So 25 to 5 lux. 25 for broilers and 5 lux for layers.
5 lux for layers.
Yes, sir. So that is one protocol is there, how much light is required. So that I keep keeping in mind.
It will change multiple times. If light intensity varies, automatically it moves.
Yes, sir, there are multiple cutoffs present.
Even in night, if some additional or
Yes, the timer is also there and multiple cutoff system is also there.
Do you see any limitation in the system?
Yes, sir, actually limitation is cost effective. We are trying to put it to cost effective, so that is one. And how it will work, that testing also we'll be doing in our coming project that has been sanctioned to us.
Okay, so I think there was any question.
Sorry, I didn't understand the basic hypothesis of your work.
Hypothesis is automation on real-time basis. Automation means we don't require any kind of human interference, and real-time basis, what is the intensity of right now in this particular room where you are sitting. So that is the main idea.
Are you trying to simulate the natural scale of light reduction? What are you basically trying to follow?
Basically, natural is natural. The automatic, the artificial one where you require human to come and put on the switch on and off. That part, no human interference. What is the right light required by that particular domestic species will fit on that particular switching device. So automatically the bulb will start as the light will be dark, or there will be darkness.
Anything with the photo period of the animal?
That is indirectly photo period only.
It's okay. That's okay. Very well. It's okay. वही तो है ना भाई. Definitely, but it's a technology. You take the technology, a good technology in just controlling the light. On light basis, you have the egg production, you have many things, milk production, all that. It can be used. The technology is a very nice technology.
Thank you. So now I would request Sunish Balhara to present.
Zero five.
05.
सब्जेक्ट में रिव्यू चलेगा, रिव्यू जाएगा, लेकिन अभी उसका IIT का होता है 12th में ही है के साथ साथ ही वो 12th भी पास करेगी। बेटी का घर कितनी दूर है? वो गाँव कल बेटी का घर कितनी दूर है आपके घर से? डिस्टेंस हाँ आपकी बोल रहा हूँ नहीं एक
आप ही कर लो बेटा, तुम आप ही ऑब्जेक्ट कर लो, उसे टाइम हमारा बच जाएगा।
बड़ी बेटी को, एक बेटी को
Respected Chairman sir, Co-Chairman, and Rapporteur, and other dignitaries sitting over here. Good evening all. My name is Suresh Malhara. Today I am presenting.
बेस्ट क्यों बेटे कह रहे हैं ना बेटा।
फर्स्ट स्लाइड ऊपर से कंट्रोल होगा।
Today I am presenting on topic developing deep learning algorithm for predicting mastitis in bovine. As we know that mastitis control and prevention is very important. Almost any bacterial or mycotic organism that can opportunistically invade tissue and cause infection and also cause mastitis. A total of 137 species, subspecies and varieties of organisms have been isolated from bovine mammary gland. A primary goal of One Health of WHO is safe animal, safe milk and safe human. As we can see from the image, last image, as we can see from the image that the Krishnamurthy et al. Study find that not zone-wise mastitis in cases that S present subclinical mastitis and C present clinical mastitis. Next. We used infrared thermography for other health assessment. Earliest detection of mastitis is key to minimize losses due to mastitis. Infrared thermography technique is a modern, non-invasive and non-contact technique which does not require any sedation and avoids traveling stress on animal. Thermal imaging is very sensitive in detecting temperature difference of less than 0.05 degrees centigrade, which is 40 times more sensitive than human hand. It also shows animal physiological state by graphically mapping the skin temperature using thermograph images and allows early detection of lesion before any clinical visibility. Can also be used for normal assessment and physiological as well as pathological status of animals. Next. Infrared thermal imaging cameras are handheld and can be put on the stand. A thermal imaging camera is an advanced type of radiation thermometer used for measuring temperature at multiple points across a large area and creating two-dimensional thermographic images. These cameras are considerably more software and hardware based than a spot thermometer. We can after getting the pictures from infrared thermography, we can analyze these pictures with the help of software, which can be give accuracy at the highest level. How we can assess these images? These are the criteria. We have to set a delta T for temperature difference. There are two forms of this type of settings: is general and specific, and based on temperature rise versus historical data. Relative temperature criteria can also be set by taking normal criteria based on temperature rises divided into 3 different categories like intermediate, serious and critical. And absolute temperature, we can set absolute temperature that we have to fix temperature and above which we can get an alarming situation. And profile assessment we can also done by analyzing the whole body temperature. Interpreting infrared data correctly, we have to keep in mind, we have to also keep in view the wind, rain, ambient air temperature, sunlight, all factor into final analysis of the data. We have to set these correction indicator in the camera itself, and then we have to fix the temperature also. Cold spots are also important in images. When comparing multiple components with infrared thermography, the one that looks different than other is probably the one causing the issues. This is a small study. We have taken samples from 100 animals, and
less than 33.5% degree centigrade, healthy quarter, and the somatic cell count is less than 2 lakh. And then the other temperature ranges, and above 35 degree, it is serious mastitis infection. Next. How to automate IRT for use in mastitis detection? Baseline thermograph. We have to prepare baseline thermographs, and it is we have to create a reference database. For example, we have to taking 3 stages: before milking, during milking, and complete milking, after milking. So these are 3 images of different categories of milking. Next. After getting the thermograph or baseline database, we have to automate this type of system for making a decision support system, and automatic system we have to create. So by going through that system, it will automatically catch the thermographs of the buffalo and analyze the data with the help of the software and give decision on the basis of thermographs and all data. Thank you.
So interesting work actually, but what actual real application point of view actually, how you are going to perform, how finally the idea for early detection of mastitis actually, how is the way actually it will be used?
So this will be used in organized farms.
Okay.
Organized farms, we have to develop a system, automatically system, one unit. It consists of infrared thermograph cameras, how to capture images, what type of angle we should keep in mind, and then we have to capture all the images and make a database, and also simultaneously we have to confirm with the help of CMT or जो mastitis detection जो टेस्ट होते हैं उनकी help से confirm करके then we have to make a database and then develop an algorithm using today's most popular algorithm and all other, and then a final algorithm we have to develop and then available in online system.
But finally, shall I understand like this actually, it will be based on the temperature differences, that is going to give us whether it's early stage or actually it is...
Yes, sir. It will detect at very early stage of mastitis occurrence.
No, I'm not an expert in this area actually, but I just try to understand.
Basically, the basic idea is that whenever there is a difference for
Yeah.
so that metabolism has increased and the energy will be, that dose will be more. As compared to the
Other
I think actually till that time actually, the disease has already set in, or the infection has already set in.
That's what they are basically
Calibrating.
With the CMT, then.
But if you look at actually overall, of course I'm using this tool and validating this tool actually is perfectly fine actually. But I find it very hard that it is going to give you a sort of major difference actually. This is what my personal...
It can be used as a diagnostic tool.
Yes.
Yeah, so of course, the study point of view is actually interesting.
Is there any threshold value for temperature rise which is available? For some clinical mastitis, they have given that chart.
Yes, sir. 37 point something. 30.
So it's all.
Yes, sir.
What about climatic variations, winter, summer fluctuations?
हाँ, सर उनका स्टडी करेंगे ग्रुप वाइज सीजन वाइज.
So you have used that camera, it's E5 or E6? It's E5 or E6?
E6 है सर. 0.
Yeah, I understand actually if you put maybe deep learning thought in this, maybe you can get the sort of major differences actually. And then most important is actually how much data you will have. All this outcome actually will be based on finally that. Then it becomes of some significance. Any questions? Yeah, thank you so much. Thank you.
बेटी बेटी को छोटी है बच्चे आगे बढ़े इसमें एक
Good afternoon to all of you. It's my pleasure to thank organizer who gave me opportunity to present respected chairman, co-chairman, reporter and distinguished scientist. So I'm going to present on behalf of Dr. Nidhi Parita a small work, piece of work of our laboratory, the efficient one-step delivery of CRISPR-Cas component in buffalo zygote by electroporation. So in the morning session also, Professor Madam has decided on the technology, the DNA technology like CRISPR-Cas. So in the recent past, CRISPR-Cas technology has well-established technology for manipulation of zygote gene at the particular locus or at the particular trait for the manipulation of gene to desired production or to desired manipulation at the gene level. So in that way, so for the transduction of the CRISPR component, there are several protocols are available, like either you can go for the cell-based transfer electroporation or zygote-based. So out of these methodology, I'm going to discuss about the electroporation of zygote-based, because all these methodology involve a lot of techniques and expensive. Apart from that, the zygote-based is quite easy, time-saving procedure and having the most efficient way. So we have a standard protocol which I am going to share with you. So electroporation is a procedure. We all know that it's a procedure. It's an electronic procedure to introduce either plasmid or ribonucleic complex into the cell or into the zygote using the high voltage electric shock. So these are some protocol or mechanism for the how to introduce the gene of interest protein or whatever so from the outer source of the plasma membrane to the nucleus or to the cytoplasm, then directly enter to the nucleus into the nuclear pore, and it will integrate with the genome, and then it will start their function. So basic protocol is there. So for the zygote electroporation, we will collect the immature oocyte, subsequently go for the maturation, then go for the in vitro fertilization. So after in vitro fertilization, we will get the zygote. So we have to standardize at what stage of the zygote we have to electroporate. We'll show you in the later stage. Then we'll electroporate it using the myostatin gene or, for example, other OCT4 gene as a proof of principle along with the guide RNA and Cas9. And then electroporate them. After electroporation, go for the culture and subsequently you have the blastose. Then check the efficiency of the procedure. Then you can check the editing efficiency. So I will show all the things in detail. So in this, so here I'm going to show you as a proof of principle, we have chosen the OCT4 is a transcription factor. So OCT4 is a supporting gene which is the embryonic development. So we have studied the knockout of the OCT4 using this protocol. So different during the standardization process, we have to choose a different voltage, number of pulse and time. So out of 20 volt and 15 volt, we found that 15 volt at 5 multiple or 5 impulse and for the 3 millisecond give a significantly higher result in terms of cleavage and blastocyst production. So further, we have checked once, we have standardized the voltage interval, time interval as well as the impulse. Then again, at what time we should and Jaguar, at what time of the Jaguar we should electroporate, either at the 8 hour of the insemination or at 10 hour of the insemination. So we have to choose two window of the time, 8 hours and 10 hours. So we found that, so 8 hours is good enough. At 8 hours, there is no cleavage, there is no blastocyst form. So we have chosen at 10 hours, 20 volt for the further experimentation. So when we compare at that standard protocol for their editing efficiency, we found that their efficiency was around 10% and around 40%. When we increase their voltage 20 volt, 5 pulse and 3 millisecond, then we got a significant higher editing efficiency up to 84%. So we found that we got the blastocyst. Further, we confirmed that blastocyst using the Sanger sequencing method for the confirmation of the editing, and we found that it's edited in the biallelic edited, and its efficiency is around 86%, and that's again confirmed with the help of specific kit for the gene protocol, this one, for editing efficiency. So in that way, we have a standard, and further we confirm their genotyping confirmation, whether this is a monoallelic or biallelic. So it was the biallelic, it was the mix. Some of them the monoallelic, some of them are biallelic, and some of the mosaic. So in that way, to reduce the mosaicism, we have electroporated at the 10 hour of the post-insemination. In that way, we found that the mosaic formation rate quite reduced as compared to the 8 hour of the electroporation of the fertilization. So further, we check the effect of that knockout of the OCT4 gene on the embryonic development. So we found that, so in terms of expression of the SOX2, nano and GATA2 gene, these genes are very essential, and they are also synergistic acting with the expression of the OCT4. So in that way, we found that there is no significant, so in the summary, so direct electroporation of CRISPR-Cas9 using a single guide RNA, highly efficient technique for causing OCT4 mutation in a, and it's a useful tool to realize the functional genome of the buffalo. Buffalo embryo with the OCT4 mutation called the CRISPR, so no difference in the blastocyst and cleavage rate formation, but the absence of OCT4 expression significantly reduced the formation of the ICM specific gene like SOX2, nano and GATA2. So this shows that the importance of the OCT4 in the embryonic development, as well as whatever the protocol we have standardized for the electroporation to the transmission of, to the transduction of CRISPR-Cas9 to the genome using the Jaguar electroporation is very efficient. So that's the piece of work. So apart from that, we are also working on other protocols. So but we have, I've shown here a small piece of the electroporation for this presentation. So we are also thanks to all the lab mates who contributing in the significant in our laboratory. Apart from that, funding agency like NSF, SCA, SCRV, ICR. So thank you very much. If you have any questions, then I'm here.
Thank you very much, actually. So, just a curiosity, actually. So, how effective actually is this protocol as compared to the protocols existing in this?
So, this protocol is around 86, showing the 86% editing efficiency. So, earlier we were using the somatic cell nucleotransfer. So, that's also, but it's a commercial, time consuming, and it's huge labor intensive, sir. So, in that way, the electroporation of the Jaguar is quite easy because that's required small only buffer and electroporation machine. So in that way the efficiency is very high, and we can do at a time 20 to 30 Jaguar at a time. But in case of somatic cell transfer, we can do one by one. So that's time consuming and expensive.
Any question from the audience?
So you show the voltages. Did you use only the coding pulse or the transfer pulse as well? You show only one pulse.
Yeah, so we have, because in electroporation, we are also having the nucleofection. So based upon the same principle, nucleofection is a patented for the Loja company. They are claiming that, so they are giving some sort of impulse which is all the either DNA or RNA directly enter into the nucleus. But in electroporation, because the plasma membrane is there, that's permeable, it's not permeable, so we are making only one pore, so that pore With that pore, your gene of interest or plasmid will be entered into the cytoplasm, and from the cytoplasm automatically it goes into the nucleus through the nuclear pore. So it's a passive way.
My point is, it's a square pulse. Does the voltage remain?
It's a square pulse. Yeah, it's a square
Or
pulse.
does it drop?
It's a square pulse.
Square pulse.
Yeah.
So usually no transfer pulse.
No, not transfer pulse. Yeah.
So what was the volume you use it for the embryos?
20 microliter. 20 microliter.
And the concentration of the lights and
1:100 nanogram. 100 nanogram guide aapne, 100 nanogram this one.
thanks.
Yeah, 1:1 ratio.
How to expedite that process of incorporation using electrophoresis?
Yes, sir.
Attempts have been made to increase the current. And in that process, we are now aware that there is leakage taking place from where we are trying to put in things, there are others simultaneously coming out of there.
Yes, sir. So that's we have standardized that one. So we are under different combination of voltage. So 15, 20.
The question is that there are certain critical elements which are coming out, and those are which are very effective in exercising an extra chromosomal effect on the genetic expression. But we talk about the text.
Yes, sir. But that aspect we have
Now,
not.
how are you going to associate these two together?
See, that aspect we have not studied yet, sir. So right now we are studying how to increase the efficiency of editing using the electroporation, macromanipulation, and cell-based electroporation.
See, at present, we don't have a system here in India. Outside, they have developed. We don't have yet a system we can take out the whole, one single embryo and then find out what's getting in and what's getting out.
No, sir. No, we do not have that much of expertise.
How are you going to do it now?
Yes, sir. We do not have that much of expertise, and we do not have that, but you are talking about that electrofusion or all the things.
All right.
So this was zona included study.
Yeah, it's a Jonah included study. So before that, yes, sir, very good question. So because Jonah is a very hard layering, so before doing that, we are thinning them using the thyroid solution, so that your Jonah will be a little bit weakened and it will be flexible, so that less harmful impact will be there because of electric impulse is there. Electroporation is an electric impulse, so that will be damage. So in that, to prevent that damage, we are using the thyroid solution, so that it's weaken the Jonah and reduce the electric impulse damage.
Okay, thank you very much. Actually, we will go to the next speaker now.
Thank you, sir.
Thank you. So next, 011, Divya. The presentation number is 011. Then we have presentation 014. First author is Shinanda. 014. 012 is there?
Before that 012 is there.
012? Yeah. Nida.
Good evening everybody. I'm here to present my topic, and the topic is ovarian response and in vitro embryo development in goats. It's a comparison of two modified superstimulatory protocols in non-breeding season. As we know, reproduction forms the backbone of animal production and productivity, and to sustain the agricultural production, we need to use all the emerging technologies, especially the modern reproductive biotechnologies. Reproductive biotechnologies increase the production, they improve the reproductive efficiency, and increase the rate of genetic improvement in animals. Superovulation is one of the reproductive biotechnologies. In this this process, actually we increase the number of the oocytes produced by the animal per cycle by the use of gonadotropin. However, the results in this are unpredictable. Sir?
Speak in the mic so that we listen what are you talking.
However, the results of superovulatory protocols are unpredictable. Our primary focus is to eliminate the variability of superovulatory responses in small ruminants. Also, the superovulated donors, they are used to the donors are used either to give the oocytes, mature oocytes, or to donate the embryos for further embryo transfer. Also, the in vivo derived oocytes, they have better developmental competence. The objectives of our study are to compare the ovarian responses in two protocols and to compare the in vitro development of in vivo mature oocytes. Our methodology included 10 animals, 5 in each group. Day 0 protocol A and group B was the traditional 18-day protocol. In group A on day 12 and in group B on day 18, laparoscope-assisted ovarian evaluation was done, which was further subsequently laparoscope-assisted flushing was done and mature oocytes were collected. These mature oocytes were then subjected to in vitro embryo production, and further they were assessed based on cleavage rate, monomer percentage, blastocyst percentage, and the blastocyst cell number. This is the Group A Day 0 protocol. In this, we used the short-term ester synchronization protocol, and after sponge removal, 84 hours after sponge removal, FSH treatment was started. This FSH treatment coincided with the emergence of the follicular wave of the next cycle, and FSH was given in 6 doses, 12 hourly in decreasing pattern. After the last injection of FSH, heat detection was done using apianized bugs, and animals were subjected to flushing on day 12. Minimum of 6 hours was kept between the heat detection in animal, and then they were subjected to flushing. In Group B protocol, day 18 protocol was used. In this long-term ester synchronization protocol was used, and the sponges were placed in the animal for 15 days. In this, the FSH treatment started 60 hours before the removal of sponge, and they were given in 8 doses but constant. The doses were kept constant. On day 16, after the last FSH injection, again heat detection was done, and subsequently on the appearance of heat 36 hours after the flushing was done. This is the flushing procedure. We used the Tomcat catheter from the Fimbre side to introduce the flushing media, and a Tomcat Foley's catheter we used from the uterine horn side to collect the media. These are the results comparing the ovarian responses between the two groups. Although the group B showed increased number of CL and N2 oocytes, but the results were not significant. The corporal lutea between the two different groups, the group A showed pale and less number of corporal lutea. These are the oocytes which were collected from Group A and Group B, mature oocytes. The Group B oocytes were better in quality. These are the in vitro developmental parameters. After the chemical activation, the mean cleavage percentage, mean monomer percentage, and mean blastocyst percentage were compared between the two groups, and in Group B, all these 3 parameters were significantly higher. These are the Group A and Group B day 7 blastocysts. We compared the blastocyst quality based on the cell number, and in Group B, the cell number was more. These are hoist day 7 blastocysts for cell number depiction. And we concluded that although both the superovulation protocols showed ovarian response with no significant differences, but the in vitro development in day 18 protocol was better, which was assessed by higher cleavage percentage, monoline blastocyst percentage, and better blastocyst quality. Hence, 18-day protocol is better suited for superstimulation in goats during non-breeding season, providing better oocyte quality and in vitro embryo development. Thank you.
Thank you very much. It's a good direction actually to work on. So, but the sample.
So it's actually an initial report. We are continuing work on it.
Yeah, maybe that is something actually which is critical for our
It's the animals we are who's there.
Sir?
How many animals we are taking? Sample size.
Sir, मैंने sample size में कम है 10 animals से 5 per group. ये initial एक report है. हम इस पे काम अभी कर रहे हैं, आगे भी चल रहा है.
So, about 5 each group? But the deviation you didn't show the data. The deviations, standard deviations.
So actually I represented the bars that as well.
Yeah, it is initial data actually which you people are going to expand further.
Yes, sir. Yes, sir.
Okay. Any question?
Would you do any repeatability?
Why did you did all this? इतना लंबा चौड़ा इसका ये बना दिया इतने injection. Why is that necessary? We have a standard protocol already available.
So we have, but in यहाँ पे non-breeding season में हमने बहुत सारे protocols रखी हैं.
जितना आपने बनाया है ये आसान बात नहीं है इसको करना.
सर हमें लेकिन
क्या उस चीज का जो हो नहीं सकेगा
सर लेकिन इसमें जो
और इसमें आपने देखा कोई एडवांटेज भी नहीं है.
सर जो हमने यहाँ पे प्रोटोकॉल सुपर ओवुलेशन के किए हैं नॉन ब्रीडिंग सीजन में उसमें हमें ओवेरियन रिस्पांस भी नहीं आया था. बस यही 2 प्रोटोकॉल्स ऐसे हैं जिनमें हमें नॉन ब्रीडिंग
What I'm saying that you have already previously done. Now you have now put it about 7-8 items that which have to go over into the system. And besides that we are talking about, you have talked of FSH also being taken over. Now I don't see any statistics doing that. Why should we be doing that? Because even without doing that, there is an response which is coming. Why should you increase the number of injections?
Sir, because the half-life of FSH is low, and moreover, if we use a single dose of PMSG, it's reported that the embryo quality and the oocyte quality are not.
That's not obvious from you have not shown anything by which that we are talking of that it increases the quality. And how will we be able to judge the quality right then just only by seeing the oocyte?
So we have checked the embryos.
Oocyte quality depends on subsequently that how it takes the shape of a fertilized zygote, and then how the embryo develops over it. That is an index of saying that yes, it was good or it was not. Otherwise, it's very difficult to say that you must, if you have experienced it or your professor might be experiencing it. उन्होंने देखा है कि जब हम when we see an embryo immediately when we collect it or we see an oocyte, that it is an index, but it is some of them they are looking very bad, but they do very well when they grow. After you might have seen it yourself. So the question is that why are we doing this much? That we are prolonging these. Why? What's the justification for that?
So हमें इसी वाले प्रोटोकॉल से रिजल्ट्स आए नॉन ब्रीडिंग सीजन में. That is why we are using these.
आप क्या हैं क्या नहीं है गर्ज नहीं है. हम ये पूछते हैं ये आपने किया क्यों? What was the इसको क्या सोच के आपने किया?
Sir, if I may add, because I have been a part of this study, so there are 2 things to the study. Probably your attention didn't go that way. So, the blastocyst, depending upon their quality, as she said, you can go to that slide if you want.
Blastocyst quality is a subsequent.
Yes. Sir,
what I'm saying is that blastocyst quality does not definitely all the time reflect what happens in its growth. I'm going before that. I'm saying that why are we putting in so much of
my point here is the ultimate index of the quality of the oocyte is the number of blastomeres or the quality or the number or the percentage of blastomeres getting out of it. That is the actual. Exactly. If you said the blastocysts, we have a significant increase, significant difference in the blastomeres of the development of these 2 groups.
Can you count the number of blastomeres?
No, of course that is one of the indexes of.
But how do you do that? You say that is the number of blastomeres.
Yes.
How can you count blastomeres?
So we do count them. So we go for Westing.
How do you count blastomeres?
So we go for Westing. That's it. We go for holistic, even the audience can count it right now. It's going to be a good thing, sir.
Whether existing protocol.
My point here is, sir, with respect, sir, if I may correct.
And we are talking about cluster land development.
Yes.
At that time, we know that cavity has already come in.
Yes, sir.
And at that time, then there is a layer. It can be 2 layers, it can be 3 layers. The question is that, do we have a method at this time? Just any method by which you can count the number of possibilities?
Of course, sir. That first thing is correct. By questioning, we have
No, my dear, listen.
Yes, sir.
When you stain it, it's a 3-dimensional, 4-dimensional picture. We are never able, as of today, go to a 4-dimensional picture of the embryo.
Of course, sir. Of course, sir. You are right in that.
I think we will discuss this. We will discuss this later. Sir, this is a very interesting discussion. Actually, we can take it just after this. So we have 2 more speakers now.
बच्चे को पता होना चाहिए अगर हम कर रहे हैं क्यों कर रहे हैं चलो thank you.
Thank you. So we have now 014 number presentation. Rishi Nanda. Then we go to 015.
Jury of the DICE or the officer and the legal those are present here. I'm Dr. Fazil, working as an assistant professor in the Department of Veterinary Physiology, and today I'm going to compare, saying something on what we have compared the fresh and frozen donor cells for cloned embryo production in buffaloes. So just going a brief about the introduction, we know that the buffalo has been one of the main important dairy animal contributing the milk surplus in the India and particularly in the South Asia, and it's a multi-purpose species, not only producing the milk but also the meat. And we have seen there is a tremendous increase in the buffalo population from the 2019 to 2022. It has increased from 307 million to 309 million in 2022. So why we need to go emphasis, and we have the belief that the reproductive technologies should come forward to have all those reproductive technologies, whether that are embryo transfer technology, artificial insemination, and other HMC cloning should be put forward. Why we need this reproductive biotechnology? The reason being that because the animal, the buffalo, there is the silent and heat and number of the various reproductive disorders are that become very unaware to the veterinarians. And if we see the efforts to clone the water buffalo by the SCNG couldn't prove fruitful. If we go through the handmade cloning, that has led simplification and the efficient production of the increased blastocyst rate in case and the pregnancy rates in the cattle. I'm not discussing here about the horse and the pig. The pregnancy rates has been increased. However, there has been the decline or the static. There has been the stagnant in the reproductive biotechnologies as it has been in the Laguna et al. In the horses. So today, I'm saying a brief about how we can compare the fresh and the frozen donor cells for cloned embryo production in the buffalo. So this is a short gist about the materials and the method. So what the cells we have taken, the study was very long, which was done in the CIRD, and it is not only the cells, but the fibroblast cells have been taken. The number of the cells varying from the fibroblast, amniotic fluid cells, and the other cells have been also studied. In this study, I have only taken the fibroblast cells. So it's if we go through this first, the A, this is a primary explant outgrowth from the tissue, and the picture B is depicting that the fibroblast cells having the 60% of confluency, cells at 40x magnification, and these are the cells that are trypsinized after 3 days of culture. As if we go through the materials and method, simply nothing that the 7 days of age, these buffalo ear fibroblasts, and the cells were cultured as per the normal protocol that we use DMEM supplemented with FBS and gentamicin and all that. And after the cells were grown at different confluencies, one more study was there that we have grown the cells at 60%, 70% and 80% of confluencies, and out of those there was another the paper has been published in that, but not in this area, that how the confluency change the blastocyst rate and the cleavage rates, and we were using these fibroblasts in the nuclear donors in the handmade cloning. This is the material and methods are continuing. These are the buffalo ovaries that we trimmed after getting from the slaughterhouse. The second picture is showing the immature buffalo oocytes of usable quality after aspiration at 10x, and after that we have matured oocytes, in vitro matured oocytes with expanded cumulus cells, and these are next photograph, the fourth photograph is showing the in vitro matured oocytes with zona pellucida, and this is after pronase treatment, and zona-free oocytes showing the pole body. So as per the protocol, all the protocols have been usable quality as well faggot and downs, and what are the how we use the pronase at a 2 milligram ml of concentration hyaluronidase treatment, and they are go with the materials and matter. These are the two demi-oocytes, two demi-cytoblasts with the donor cell, if you see the fifth picture, and in the sixth picture there is a demi-cytoplast with somatic cell, and this is a fusion after the last photograph after 2 hours of fusion. Coming to the results, we have seen the comparison of the fresh and frozen donor cells for cloned embryo production in the buffalo. In the fresh fibroblast, the number of the HNCs that were cultured were 33, but the number of the number of the cytoplasms were 286, and after that the cleaved embryos were 23, and the blastocysts that reached the blastocyst stage were 6, and in the frozen fibroblasts, if we see the cleaved embryos were 18, and the blastocysts at the from the frozen fibroblasts that reach after this at the blastocyst stage were only 4. So here we can say from this presentation that there were non-significant differences between the blastocyst and the cleaved embryos that produced from the fresh and frozen fibroblasts. And these are the representative photographs of the handmade cloned embryos at the different stages of development. So the conclusion is like that frozen, we believe that the frozen cells that are at much more higher risks, and they cannot be the source of the handmade cloned embryos. Second, there are the alternative sources that we have the concept that the fibroblast cannot be used as a source of the handmade cloned embryos. No, but the efficiency increases. The efficiency is overall the same in different types of cells, as we can conclude, but it varies from lab to lab variation. But one of the important things that cumulus cells have been seen to have better percent in blastocyst formation, and we can also conclude that in agriculture and the rest of the cloning is employed to create the pest resistant plants. Cloning is also used to create the transgenic animals, and we have proteins and vaccines and antibiotics, all are produced by the process of cloning. This can include the DNA copies, cells, organs, or even the complete animal aid using cloning. And if one more thing, what my personal reason is, someone's animal or a favorite pet die, we can have that animal back in the form of the cloning. It can also use to develop the human drugs and medications such as antithrombin. Moreover, the cloning can be used to produce and culture stem cells and be used for the treatment of many human diseases. Thank you.
Thank you, Dr. Fozia. Actually, just a suggestion to add to your study. Yeah. I think if you take 3D, this is all in 2D. 2D means actually just you have the isolated cells.
Yes, sir.
That's one type is just the flesh and another is the frozen one.
Yes.
But in the third one, actually, if you just have the frozen, but on a matrix, on a 3D matrix, actually,
yes, sir.
that is sort of compared to it. I don't know what will be the outcome, but I feel actually that outcome will be much better.
So that we will do this for
It's just only actually,
yes,
now the 3D materials actually, because that provides you actually the natural environment. Yeah, so that perhaps actually is the best way. I'm just only telling this thing also actually by my own experiences actually. So we have done that type of thing actually. Freezing the cells actually, which normally actually all the cells you freeze mammalian cells. So but if you freeze them like on the support, that is definitely a better outcome actually overall. In fact, we have the same thing actually, the cells which you get transported, for example, in a sort of wild, in the media. So you have a lot of damage in that actually, but the same thing is transported actually on the support.
Yes, sir.
So then that support actually helps actually more viability in that. Yeah.
Yes, sir. Thank you, sir.
Donor cells is getting.
Yes, sir.
The donor cells. What are donor cells from? From where?
Yes. So we have the less than 6 days of it from that Murrah buffaloes at the CIRB.
No, that. So this is not a difference in donor cells. Donor cells were the same. One was frozen, another was.
Yes, sir.
Isn't it? So how do you say that is a donor cell? It's a process. It's a process.
Yes, sir. So because
you put the title as donor cell.
Sir, because
it was a donor cell. They are the same.
Sir, because when we go for sir, when
is that you know. You should see that in the end what you have done. You've gone all the way down. आपने कहाँ से कहाँ ले गए हो? मुद्दा क्या था? What were you trying to look and what have you concluded?
Sir, we have
a slide अपना दिखाइए जरा. Number 3 पढ़िए.
तो एक टेबल आप दिखा रहे थे
जी सर,
तो वहाँ पे सर को दिखा दीजिए वो वाला टेबल है, वहाँ पर कंक्लूजन आपने ड्रा किया।
फहीम प्रेजेंटेशन फिर से लाओ।
I'm constrained to say this because the bright people who see they should not do this type of, or they should do it with caution. Next करिए हाँ
yes.
Protein, vaccine and antibiotics are all produced by the process of cloning. This can include DNA copies, cells, organisms, or even complete animal using cloning. ये कहाँ से आ गया? ये क्या किया आपने? ये कहाँ से आ गया?
Yeah, I know there is some mistake.
Futurist बात कर रहे हैं।
मैं तो कहता हूँ कि in agriculture cloning is employed to create persistent cloning
So, okay, it may not be that handmade cloning, but it is a cloning. So, cloning word is there.
इतना कंक्लूजन आपके काम का नहीं है, आपके वर्ड है लेकिन आपके काम का नहीं है।
cells. So basically that you say that this is a blastocyst cell, right?
Yes.
First say frozen blastocyst cell.
Frozen
to generate
Fibroblast.
Fibroblast, sir.
आप कहते हैं frozen blast cells.
नहीं, sir, fibroblast है.
Fibroblast, वही ना fibroblast cells.
Yes, sir.
That's what I mean. आप पब्लिकेशन देखिए कि आपको क्या जरूरत है ये कहने की. फाइब्रोब्लास्ट से तो सब ऑलरेडी है.
सर मैं
I couldn't get it.
Where are we going to
चेयरमैन जी समझाएगा ये क्या है बात करना क्या चाहते हैं.
नहीं, I didn't get, sir.
मैं ये कहता हूँ frozen fibroblast cells may provide an alternate source.
Yes.
अभी इस सेंटेंस में क्या है?
Sir, because there is the concept.
May provide.
Yes, because sir, I like to add here that in handmade cloning we say
It
that.
has been already shown that they do provide.
No, sir.
We are uncertain about it.
No, sir. Sir,
The
we have.
publications are already available. We show that yes, you can make from them.
We can make, sir, but there are reports that they say the fibroblasts are not good for handmade cloning. Only the cells that have the less telomerase activity and the more cells that like the cumulus cells, the cells that are derived from the Watten's jelly, they have the better competence in expression and incrementing remodeling genes as compared to the fibroblast. So we have the concept when
Fibroblast cells, right?
yes.
Now there is a definite definition for a fibroblast cell. And you are saying that frozen fibroblast cells may provide. That means you are not certain. But I am certain that yes, they provide.
But with less competence.
But why should you be talking about certainty when we have already established certainty? That's what we're calling.
थोड़ा लिखने में चेंज करना है बाकी.
Okay, thank you.
Thank you. Anybody agree with that?
I think nobody is here.
Nobody is there. Nobody is
You
there.
can conclude.
Anyway, thank you very much actually for this wonderful activity which also generated a lot of discussion. Thank you for all the speakers actually which we have on this session. Yeah. So any announcement you have?
Yes.
I would like to call Dr. Riaz Ahmed Shah to present a memento to chairman and co-chairman, Dr. Ashok Kumar sir and Dr. P.S. Yadav Thank you, sir. I would like to call Dr. G.S. Gautam, sir, to present a memento to Dr. Dharmender, sir, and Dr. Nitin Kumar Gadde, sir. Nitin Eknath Gadde, sir. Thank you so much. I think our sessions are over. Now there is general body meeting in this hall. Sir, what time it should? General body meeting. Sir, it is 6:25 right now. Sir, I think we should start by 6:30 or sir, general body meeting.
Meeting was about other sessions. We don't know the stats of this session.
Sir, our poster session is already over. Another session I will call.
Where about that then?
Then sir, we will have a break of 15 minutes.
General body meeting. क्या बात? ठीक है sir. आप सब.
We will have a break of 15 to 20 minutes.
We don't have the tea over here, but they can have a break.
Sir, we will have a break of 20 minutes. We will start by 6:45.
नहीं, sir can.
Sir, it is 6:25 already.
टाइम खराब करने की क्या चाय पियो खत्म करेंगे
सर दूसरा सेशन अभी पता नहीं है कि वहाँ सिस्टमिक सेशन जो चल रहा है ना सर, सिस्टमिक फिजियोलॉजी
चाय बनी हुई है या नहीं आई,
पता करेंगे सर।
मतलब मेरे कहने का